had a 3 years funding period, followed by another year budget neutral extension, and I guess we are well on track. I have to say that, or I have to acknowledge Tim Alsop, of course, from Lucentis Pfizer, who started these efforts. So I had the pleasure to step in as a coordinator only beginning of this year because the Lucentis site was closed. And since then, it is my pleasure and some pleasure, I have to say, to act as a coordinator because there is a lot of paperwork involved. And you will see why in the following. I hope that everything is set. Yes, here we are. So I'm supposed to give you a brief overview on what EBiSC was or is and what the future of EBiSC is going to look like. And because I'm from the FPIA employee, I'd like to give you a bit of an impression of what the FPIA group or the FPIA company think such an EBiSC or IPS repository should look like, or what we are mostly interested in. And as a disclaimer, I have to say I'm an employee of Janssen, so this is a disclosure. And I can give this sort of an introduction only from the Janssen perspective. And to be more specific, it is from the Janssen neuroscience perspective. So I'm working at Janssen in Belgium as a group leader, scientific director in the neuroscience department, and we are mostly interested in Alzheimer's disease. And you can probably see from this industry perspective that it is somewhat neuroscience focused. So what is our perception about the iPSC technology? I mean, we are experiencing since a couple of years a boost in the IPS technology. We are bombarded as the FPIA company with requests to collaborate. There are more and more IMI projects that are in the process of being funded or that just started, or this one which is now closing down, that have a significant focus on the IPS technology. What I'd like to underline here is what we think is needed. So first of all, we still are under the impression, or we think that IPS are a preclinical research and development tool. So we are not yet at a stage where we consider iPSC to be a therapeutic. I know there are a couple of activities ongoing for macular degeneration also, where iPSCs are being developed as a therapeutics, but in the neuroscience area, at least where I come from, this is mostly neurodegeneration, we consider iPSC as a preclinical tool. We would like to use these iPSCs to study the pathophysiology, and for studying neurodegeneration, we need to have access to neurons, astrocytes, microglia, and all human origin. And you can imagine that it's pretty complex to get hands-on primary material for these cell lines. So therefore, the iPSC technology is of interest to us. What for? So first of all, we would like to understand the disease phenotype. We would like to then set up screening assays. So we need large amounts of these iPSCs in order then to study any phenotype that are detectable in these cells and then reverted by small molecule approaches, for instance. And this all to help bridging the gap from preclinical tools, mouse models whatsoever, and these have their limitation, especially if it's down to neurodegeneration, to then human studies. So another important point is, I mean, we are pharma. We would like to use these tools in order to deliver into project portfolio. So we have a certain question that we would like to answer. So for instance, how to stop tau pathology, and we would like to use iPSCs to answer these questions. It's not much about basic research. We would like questions to be answered in order to progress our portfolios. So we have not the time to study and develop assays and technologies in order to make them a tool. So we would like, in an ideal world, to take a developed IPS line, IPS differentiation protocol, to then forward it to our laboratory and start experiments. And the last bullet point is something that I collected from the FPIA partners within EBiSC, and it basically says what I have alluded to already. We have the readout from iPSCs in some discovery projects, but so far, at least in our shop, iPSCs have not yet been in the position to drive decisions. but I hope that in future we will come to this. So what is our expectation then? So what is the must? What does the IPS technology for at least my business need to deliver? Most importantly, it needs to be reproducible and robust. And in an IMI, we started in EBiSC, we started cross-laboratory experiments where we sent IPS lines out and where we asked the different partners to use the very same differentiation protocol, the very same treatments, the very same readouts in order to then assess, do we get the same data? Do we get the same results? I think this is absolutely important because the reproducibility of publication also is a concern to us, and I guess to the scientific community in general. We would like to have confirmed disease phenotypes in these lines. So say if we have a IPS line, for instance, from a patient suffering from frontotemporal dementia, for instance, we would like to have a phenotype that we can reliably detect in these lines in order then to subject these lines to phenotypic screenings, for instance. And this needs to be reproducible again. In an ideal world, we have documentation attached to these lines. So we have standard operation procedures, we have the differentiation protocols that we can access when we, for instance, purchase an IPS line from the EBiSC repository, for instance. Important as well is that access to patient records, clinical data, the whole genome sequencing, for instance, is available as well. So, I mean, if I'm studying, for instance, a line from an Alzheimer's disease patient, I would like to know, are there SNPs that have been linked via genome-wide association studies to Alzheimer's, yes or no? What is the ApoE phenotype, for instance? And I've seen one poster outside where, which claims that this information is partially available already within EBiSC. Very important for us is the 4 bullet points, is the freedom to operate. When we started with IPS work at Janssen, we were approaching small biotechs, university groups, and asking for iPSCs. In order then to get these iPSC into Janssen, this triggered a huge amount of negotiations and contractual legal work. So it is not easy to just approach someone, ask for the iPSC lines to be sent to Janssen, we start working. There are often not the correct consent forms, the patient consent forms attached to it. Tech transfer offices seem to be sometime thinking they can add reach through claims to these collaboration agreements, and this is all not something that we would like to have in place. We would like to use these cell lines and use the data that we get for research and development purposes. I'm not talking about commercialization or so. Related to this is the ease of access and the deposition. Again, the legal and administrative burden is often prohibitory to get IPS lines in-house. At a certain time and point, we were just saying, okay, leave it. The work attached to it is more than we would accept. So it is not matching what we expect as an outcome. Of course, we are following the IPSC offering from the outside, and this needs to fit with our internal research and development pipeline. So we are flexible with regard to tools. I mean, as you all see the CRISPR-Cas technology evolving, we are now switching from patient-derived IPS line to gene-edited cell lines because we then have the proper controls. And this is something that we have on our radar and needs to be monitored constantly. And at the end of the day, we need to deliver results. So in my goals and objectives, for instance, it says you have to deliver X at the end of 2017 or 18. So an iPSC technology needs to deliver into the projects within a commercial entity. I mean, this goes probably without saying. I'm happy to take any question if there is one already now.
How do you deal with informed consent? So there's a big debate also on the level of ISSCR on induced pluripotent stem cell, how we should internationally deal with informed consent forms. So we had something translated now in Germany, and then the lawyers in Germany say, oh, this is a big mess because you get into translation.
This is
so how do you deal with that?
I'm not dealing with it because I'm not a lawyer, but
Well, you know, but
you're right. This is something that has been dealt with in EBiSC. So we had one work package dealing entirely with the legal aspects, the bioethical aspects. How does an informed consent need to look like? Can we harmonize it across Europe, which is difficult indeed. So currently there is an informed consent document available within EBiSC, which we, for instance, also send to STEM Bank or to other entities or collaborative projects that make use of these informed consents. And I guess it's fair to say this gives a relatively good coverage of what's possible within Europe. But again, I'm not a lawyer, so that's all I can say to this. But you're right, this is difficult, especially if it's down, for instance, to adding whole genome sequencing data to these IPS lines. And this is especially difficult in Germany. But this is something that needs to be worked on. You're right. So now a bit to the history. EBiSC started in January 2014. It was kicked off or initiated by New Centers, by Tim Alsop. And it was supposed to run until 2016, and 2016. The rationale was pretty clear, and it's all, again, related to what I mentioned before. The IPS landscape is fragmented across Europe. There are lots of small biotechs, universities that have iPSCs developed, and it would have been beneficial if we can centralize this offering into one banking entity. And what comes along with these fragmented spectrum is that the quality of the IPS cells is quite diverse. Depends on whom you approach. The identity of the IPS cell lines is not guaranteed. There is no phenotype information attached to it. It took a lot of time to get hands-on because of this legal aspects, and often we were stuck in discussions and as to what extent we can use these IPS cell lines for our internal research. And this is not only true for commercial entities, I guess this holds also true for public entities. And therefore, the initial goal was to establish such a European-wide repository which takes over most of these questions here, and by this minimize duplication efforts. So let's say there is an AD-derived cell line Lilly is, for instance, interested in, they start in negotiations with a certain group. If Pfizer is interested in, they do the very same thing, and so on and so forth. If this has been done once, and this cell line is deposited at EBiSC, you go there, you order the cell line, you can start working with it. So this is one negotiation, that's it. This would make use of a great use of synergisms across Europe. And importantly as well, and this was one of the goals of the IMI, they are closely following sustainability questions, that this banking entity at the end of this funding period is self-sustainable. So all the IPS lines that have been generated are available via this IPS repository also beyond the runtime of the project. So to the specifics, the EBiSC project again was funded for 3 years and then experienced a budget neutral extension until the end of this year. So this is his last EBiSC 1, let's phrase it this way, meeting, I guess, and December 31st, EBiSC is history. Just the formal collaboration agreement, but the IPS lines will be available beyond this, thanks to ECAC and Fraunhofer, who are willing to secure the lines in the repositories. It was a huge project. It had 26 partners across Europe, FPR companies and governmental authority, universities. And the initial goal was to collect up to 1,000 IPS cell lines that will be available via the central IPS repository. We have established 2 facilities. The one is now ECAC, and the other one is located in Germany at the Fraunhofer, who are running the mirror bank, which I guess you can agree is important in order to secure the survival of these lines. Another work package was dealing with the proof of concept. How can we use these iPSCs in discovery projects? And this relates to what I just mentioned before, sending out IPS lines to different laboratories and then running the very same protocols in order to prove that we are in the position to having reproducible differentiation protocols and readouts in place. This is a consortium. You can see it's spread across Europe. There's just one comment I would like to make. You see, for instance, the Spanish partner, and this is going back to what I just mentioned with access to lines. When we were starting with the iPSC work, we were planning to get IPS lines from Spain into the central repository, and this is not foreseen in the Spanish legislation. So we had to stop these efforts, and this relates back that it does really make sense to go for an Europe-wide harmonized repository and agreements. So, what is the core business of EBiSC? What was the initial goal? We have the research projects all over Europe, and we have then other researchers that might be interested in getting hands of these IPS lines. So then the goal was, okay, let's establish this IPS repository, which takes over IPSCs generated and makes the access of these IPSC lines to other researchers easy. And this via the central facilities, being the central or the mirror facility. The consent forms and contracts need to be harmonized. This was the goal, in order not to renegotiate again and again access to lines. Common standards and QC processes for all of these lines. So if you buy a cell line from this repository, they have undergone the very same QC procedure. There are precise SOPs for each step established, which makes sure that you get what you want to have in-house. A data management system attached to these lines, storing, for instance, differentiation protocols, sequencing data, phenotypes, and researchers that make use of the iPSCs are actively approached and asked to deposit data they generate with these lines into this information management system. I mentioned already the catalog, which is in the process of being larger and larger currently. I don't know where we stand with the cell lines, but a couple of hundreds already available. And there are ongoing discussions to internalize, for instance, cell lines that have been established within the IMI STEM Bank project. And then, of course, validating these process by phenotyping of selected lines. This goes back again to the reproducibility of what can be achieved with these lines and these resources. So this is now where we stand currently. But again, EBiSC ends this year, and the question is, what's then? Now, this goes back to the question of sustainability. IMI is very firm with making sure that everything which is established with an IMI project is sustainable. So it does not mean we have a project funded for 5 years, the project closes down, everything is lost. So we are actively asked to make sure this does not happen. So for this purpose, and I don't want to go too much into history, how EBiSC 1 was planned and executed, we have now started to discuss with IMI and the FPR companies to launch a follow-up call in order to make the EBiSC repository, or the European banking entity, which is currently being taken over by ECAC. This is a legal custodian of the IPS lines, is able to further develop these lines to reach this self-sustainability. So in the current call topic text, which has been published recently, this is a draft. The final one is going to be published this month, I guess. We have 10 FPR companies identified who are interested in a following up project, and this underlines how important we, from the industry perspective, value such a repository. The financials have been agreed upon, the budget is close to be fixed, and we aim for another 3 years of duration. The focus of the new consortium, this is laid out in the call topic text, is the banking, distribution, and expansion of lines. So this is really the core business of an iPSC repository. We would like to ask a new consortium to add another flavor, which is the bulk production of lines. So again, in one of my first slides, I was saying, in an ideal world, we take something from shelf, we transfer it to our laboratories, and then start screening. This means, for instance, we would like to order 1 billion cells, NPC state, which we can then plate and start screening in order not to invest our resources in such an effort. Currently, the core therapeutic areas we are talking about is neurodegeneration, diabetes, cardiovascular safety, but the call is open for other indications as well. Again, the final call topics is currently being launched, is with the European Commission currently for final approval, and I hope, if everything goes right and fast, that the new activities can start by the end of 2018. So this is something for the future of and potentially EBiSC 2. So what's the benefits? I mean, you can read through this pretty busy slide, but it's basically what I already mentioned in my talk. We will establish and derive new lines. They will be constantly integrated in the new IPS repository. One thing the new bank needs to consider is how to incentivize for instance, groups at the universities to donate or to give or hand out their cell lines in order those to be integrated. This is something which has not been really successful in the past. We have, and this is important for everyone, we have a high quality backup. So if our cell lines are contaminated, we can always go back and order new cell lines, and we can be sure, because of the QC process, that the cell lines are the very same that we ordered in the first instance. This is what I just mentioned. We hope that the new consortium will be able to produce bulk quantities, so we can order not only a vial, but let's say 100 vials in order to accelerate our internal processes and research development activities. I appreciate that the training that has been offered by the current EBiSC consortium, all the workshops were pretty successful, and I guess there is a lot of online tools available in order to train newcomers to iPSC technology. And this is also important to harmonize how the process, how to deal with a certain cell line, how to differentiate, how to put them in culture, and so on and so forth. And I guess the new EBiSC will continue with what this EBiSC project has been prepared and accomplished. Okay, sir? No questions?
So how is the liaison with the other banks, for example, those in the US? Because there seems to be a lot of overlap and duplication, and for example, Richards seems to have all the distribution issues pretty solved.
I'm aware that there are lots of IPS banks in the United States. In an ideal world, the European, so this EBiSC or the new EBiSC is going to join efforts to exchange lines and to make use of synergisms. It's basically something that this is my perspective. That's what EBiSC aim for, is to make use of synergies in Europe with regard to IPCs, and I don't think that it shouldn't be possible also to do it worldwide. But I guess EBiSC is leading with regard to the quality control of the IPS lines, and I guess other IPS repositories need to adapt to the quality standards that EBiSC is offering.
Although the NIH NCATS program has got most of it going with robotics, which seems to go very well, and as does the New York Stem Cell Foundation. So there are quite good banks being developed elsewhere, and I wondered whether the dialogue was ongoing or not.
I have not been involved in this dialogue, but one of the goals of the new call is in fact to reach out to other repositories, to make sure they are talking to each other and there is not duplication of efforts at the best what's possible. I mean, when I was starting some IPS work at Janssen, there were calls out They were saying, okay, we will derive 500 IPS lines from AD patients, and we will derive another 1,000 from Parkinson patients. No one in the world can characterize these lines. There's no way into it. So it needs to be a rethinking, and I guess the characterization of the IPS lines and also the reach out to other activities and collaborations is of utmost importance. I'd like to add that, for instance, one goal of this new call is as well to survey what's going on in Europe. I mean, there are so many projects that develop iPSCs that are funded by the European Commission or by governmental funding bodies. And all these lines, they vanish, and within the universities they are not accessible, although they have been developed with public money. And it does definitely make sense to get these cell lines into the repositories.
Well, maybe along those lines, how do you deal with cellular therapy, possible cellular therapy coming after using iPS cells for that? Is there an idea to develop a protocol how you can get an iPS cell line which is non-GMP into a GMP setting so that it could be used also in a clinical cell therapy setting, which are the other side of the banks, right? I mean, what is built up in Japan in the moment is really banks which go into cellular therapy, right?
Yeah, cellular therapy is not in the focus of this, was not in the focus of this effort, and it won't be in the focus of the new EBiSC exercise. I agree it's important, but this probably is something for a new call to deal with this, because there are quite some challenges attached to this as well, which cannot be covered here.
Sorry, let me just address the question. The CERM collection, which Cellular Dynamics helped put together, we kept back-banked PBMCs. So if anybody wanted to go make GMP lines, those materials are available from the California Institute of Regenerative Medicine to do so. To take the lines that we made and put them into a GMP facility is not something the FDA is allowing at this point, but you could go back to the original material if you wanted it.
And there could be protocols which allow you to put an iPS cell back into a GMP.
There are protocols. The FDA, I think Christine was with me at NCATS. The FDA and the NCATS people were there talking about how you could move those materials. But the original donor samples are still accessible, and they were kept because the idea is that we may not be making pluripotent stem cells in exactly the way you might want to make them going forward in the future, and you would need that resource material to access.
Further question? Okay, well, thank you very much.